Hey guys, can we please turn on to chapter 3, please? Thank you. Okay, so now we have finished talking about, remember that model that we had? We beginning from at the very top. We had a population. The population is made up of organism. The organism is made up of organ systems. Organ systems are organs, and organs are made up of tissues, and tissues are made up of cells. And then below the cell, that's where we had to learn about our chemistry of life and these proteins and these sugars and the lipids and the nucleic acids. And now we have finished the cell. So now we have finished the intracellular life. Now we're moving up. We're now going to go to see what's happening at the level of the cell form and function. Okay, so now what is cytology? Ology is the science or the knowledge of or the study of. So that's the study of the cell. It is the scientific study of cells, and there are generalizations that we need to know about the cell theory. So what does the cell theory says? That life, the basic unit of life and the basic unit of death is the cell. We are alive because our cells are alive, and we are dead because our cells are dead. We are healthy because our cells are healthy, and we are sick because our cells are sick. And then we must believe that come cells come from other cells, that cells come from cells that are cells come from pre-existing cells. So cells don't come out of nowhere. There has to be a cell. So we are here because our parents were here, our parents were here because our grandparents were here, and so on and so forth. Same thing with the cell. So if you had an injury, then the cells who are around the injured tissue, after inflammation, the injured tissue is going to be removed, and the necrotic debris is going to be taken away. Then that is going to induce the differentiation. That is going to induce that's going to induce the difference in that's going to induce the proliferation and differentiation of the new cells. So new cells arrive from pre-existing cells, and all organisms are composed of cells and cell products. So everything that we have in an organism is either itself a cell, or it came from as a product of some as a cellular product of the cells. Then the cell is the simplest structural and functional unit of life. The cell is the simplest and structural unit of life, and an organism's structure and function are due to the activities of the cells. An organism's structure and function are due to the activity of the cells. So the cells come only from pre-existing cells. So we have different types of cells. We talked about this the first day of class. We said that how come we have a neuron, we have an islet of Langerhans, the cell of your pancreas, we have a hepatocyte, the cell of the liver, we have an osteocyte, the cell of the bone, even though they all have the same DNA, but they look different. Remember, we said that the knife and the hammer are both different, even though are both made from steel. So the knife is made from steel, the hammer is made from steel, but the knife, it can cut. The hammer cannot cut. The hammer can hammer things down. So the reason is because the structure is different. The knife is sharp. That is what allows the knife to do the function of cutting. And the hammer is dull. That is what enables that is what enables your your hammer to do its job. So these cells, the bone cell, the osteocyte, the nerve cell, the neuron, the islet of Langerhans, they look different because they have different functions. How did they achieve that? What led them to become different? Obviously, because they have different functions to do, so they have different structure. But how did they achieve that? How did they manifest that? How did that happen? They all have the same DNA, so how did that happen? Yeah, so how did we achieve the specialization? I'm sorry? Okay. The what? Transcription. Excellent. Of what? Yeah. Okay. So we have the same cell. Okay. And the cell has, today is throwing markers in the trash can. Okay. So we have the same cell. It has it has the DNA. But this cell can give rise to a neuron. It can become a hepatocyte. Hepatocyte is what kind of cell? Of the liver. It can become a hepatocyte. It can become a bone cell, an osteocyte. It can also become a cardiac myocyte. But even though they have the same DNA, so what led the cell to go down that path? Your friend said a very good answer, that is transcription. But I need more detail. What helps me? What do I try? How do I get these different cells? Okay, there is a region in the DNA. If I transcribe it, I will get a neuron. That's region number 1, will give me cell 1. Region number 2 will give me a hepatocyte. Region number 3 will give me an osteocyte. Region number 4 will give me a cardiac myocyte. So the region that I transcribe to make me what I want to be, that is called what? That is called what? Euchromatin. That is the euchromatin. I'm repeating the same thing over and over again, and people are still not picking up. You don't remember euchromatin and heterochromatin? That's the first day of class, guys.
Not with us. No. You have many classes, so you're confusing us.
No.
No.
We said that we have different regions of the DNA. As your friend said, we transcribe them. We didn't do the central dogma of biology. The central dogma of biology is when we go from transcribing DNA into mRNA and from mRNA into proteins. So we transcribe one region, then that region is going to give us a particular set of proteins. We transcribe region A to get mRNA, then we transcribe mRNA to get proteins of the neuron. That is known as euchromatin. This is the one that we transcribe. Heterochromatin is the region that is not transcribed because I don't need those. So then they are heterochromatin, everything else, that is euchromatin. If I want a hepatocyte, I transcribe this region, that is known as euchromatin, everything else is heterochromatin. If I need that region, I transcribe region number 3, that will be my euchromatin, everything else is heterochromatin. If I transcribe this region is known as euchromatin, everything else is heterochromatin. Okay, I'm gonna go in the recorded lecture and I'm gonna send it to you guys. You see, because you said I didn't talk about it. Okay, in this class. The heterochromatin, euchromatin, this part, that I talked about that. But I'm gonna send you guys the video because you said I didn't talk about it. Okay. You don't remember, but you said I'm confusing you with my other classes. I teach all the classes the same content, so I wouldn't leave you guys out from this is the center of what we're doing in anatomy and physiology. Okay, so these cells. So now we talked about these different types of cells. So the cells have a traditional type of tissue. That's the squamous, the cuboidal, the columnar, the polygonal, the stellate. These are different types of tissues that have a different structure. So the squamous type of cells, they represent for me a cell that is like an egg sunny side up. So you see the nucleus is really prominent at the top. It's really prominent showing at the top. And so that's them. So there was a question on the exam that you had to see all of those, but I deleted it. So the question was, you would have 1, 2, 3, 4, 5, 6, 7, 8, 9. You would have these 9 with no names. You see the names here? Squamous, that's cuboidal, that's columnar, that's polygonal, that's stellate, that's spheroidal, that's discoidal, that's fusiform, and that's fibrous. So you had to know which one is which. So I removed that for now. But for the midterm, you're going to come back and you're going to see the same figure. And when you say, which one is discoidal? You say this red blood cell. Which one is stellate? These nerve cells. Which ones are fusiform? These are spindle shaped. These are your smooth muscle cells. Which ones are squamous? These are the cells that are found in your esophagus. So these are the common cell types. So most human cells are about 10 to 15 micrometers in diameter. So this here is an example. So you're not responsible of knowing everything on the slides, obviously. There are 150, like right here. I'm not going to ask you on the exam, what is the usual length and width of the cell? No, this is just general information that could help you understand some more. So don't ignore it, but do not dwell on it to memorize it. Like, for example, but every picture, every figure, you have to know that one. Like, what does this figure tell you? This figure tells you that this cell has a diameter of 10 micrometers. If I double it, how will that affect my cell? So now, if I double the diameter from 10 into 20, what happens? The surface area is going to increase by a factor of 4, and the volume is going to increase by a factor of 3. So that means the distance. So now this room, imagine this room is a cell. This room that we are in is a cell. If I multiply it by 3 times, now the molecule, instead of going from here to the nucleus in 1 millisecond, now it's going to need 3 milliseconds. That's too late. Then the cell will die. So the proportions of the cell have to be in proper, exact proportions to help us. They have to be in proper, exact proportions to help us have a cell that is functioning to sustain life. So this one, we need to know that there are improvements in the technology that allows us to see the cell. We said back in the day, it was all gross anatomy and physiology. What you can see, that was all the anatomy and physiology right there. That was your cadaver anatomy and physiology. But when they discovered the microscope, they said, no, anatomy and physiology is not only gross at the level of the organism and the tissue and the organ. There is anatomy and physiology at the level of the cell, and that is known as that's known as histology. So now when we study anatomy at the level of below the microscope, things we cannot see with the naked eye, then that is known as the histology. So that shows you the difference between magnification and resolution. What is the difference between magnification and resolution? Magnification is how close are we, like how magnification means it's our ability to see, in finer detail, while resolution is when we see in finer detail. Magnification is when I can see small things that I could not see before, but resolution is when I can see in fine detail. What's the difference between magnification and resolution? Excellent. Is when I see in like very fine details, the intricate things. What about magnification? I'm sorry? Yeah, you see the small, you see magnify, you see things are bigger. They are small, you see them, you make them bigger to see them. This is very important now. What are the basics? So now if we want to study the cell, so what are we going to do in the cell? You see somebody, when you tell them, study all the New York City, what are they supposed to do? Are they supposed to go memorize all the name of the counties first? Name all the counties first? Then you go up, you know, the major boroughs, and then you know the state, or should you begin first? Should you begin little and then go up, or should you go up then little? Yeah, from big to small. So you have to know the continent, North America. Then you have to know the country, the United States of America. Then you know the state of New York. Then you know the boroughs. Where is this? Manhattan, right? Manhattan. And then you go is Harlem. But imagine somebody who knows Harlem and Manhattan, they have no idea what America is and what's the North America is. I guess some people are like that. So when it comes to us studying the cell, we begin big from outside into the inside. So the cell, we divide it into three very big components. The cell membrane and everything that's in it. Number one is the cell membrane and everything that is in the cell membrane. Second of all is the cytoplasm and everything that's in it. And lastly, the nucleus and everything that is in it. These are your three major components of the cell. Then you can take the plasma membrane and dissect it into little pieces. Oh, it has phospholipid bilayer. Oh, it has hydrophilic heads. It has hydrophobic tails. It has embedded transmembrane proteins. That's later. But for now, I need you to know the three big things, and then we hold each one, we dissect it to pieces. So what's the first big thing? It's your cell membrane. That is what gives you your dimension. That's what gives you your capacity. So the cell is surrounded by a plasma cell membrane. It defines the cell boundaries and is made up of proteins and lipids, and its composition can vary between regions of the cell. The cytoplasm is within the cell. So that's the other region. It contains the organelles. Some people thought the cytoplasm is the liquid part. No, the cytoplasm is the region. Is the cytosol. That's the liquid, like egg yolk. Egg white, not egg yolk, the egg white part. That's the liquid jelly part. And it has the cytoskeleton. That is the mechanical proteins that help the cell achieve its function. Like when the cell wants to move, it pushes the cell to start crawling. The cell can crawl because of the cytoskeleton. And there's inclusions. These are like warehouses where we store foreign particles and we store food in there too. And clear gel called the cytosol, also known as intracellular fluid. That's really important. What is part of the cytoplasm that helps the cell walk and crawl? What is that? The cytoskeleton is the skeleton, the scaffold that allows it to move. Extracellular fluid, ECF, extracellular fluid, is located outside of the cell. Extracellular fluid is located outside of the cells. So the extracellular fluid includes any fluid outside of the cells, including tissue interstitial fluid, blood plasma, lymph, and cerebrospinal fluid. Now that's outside the cell. So we need to understand what's happening outside the cell, what's happening inside the cell. Why is that important? Because you need to know the electrolytes. Is there more sodium outside the cell than inside? Why is that? Why are we so concerned with what's happening outside? Are we only concerned with what's happening inside our school? Do we not care what's happening outside? Is it raining? Is it cold? Is there homeless outside? Is there dangerous people? Why is there public safety down there? Why don't we just live here? Why are we so concerned with having a cell membrane? We are concerned with outside too. So we don't just study the cell. We study outside. Because outside is bacteria, outside is dangerous stuff, outside is the electrolytes. And we're going to learn that in this membrane, you have embedded transmembrane channels. These channels open up and allow the influx. We don't say entry, that's layman language. We say the influx or the exit, the efflux. The extracellular fluid is located outside of the cells. The extracellular fluid includes any fluid outside of the cells, including tissue, interstitial fluid, blood plasma, lymph, and cerebrospinal fluid. Okay, so what does that show us? These are the structures of your cell. And you obviously saw there was a question on the exam about that. So you have to know these structures and their function. That is my nucleus. And then there's the nucleolus. And then between them, you have and then above them, you have your nuclear envelope. So the nuclear envelope and the cell membrane on the organelles, all of them have membranes. This is the Golgi body. It looks like pita bread stacked up on top of each other. You know the pita bread? Yes. Which one is what? The envelope is this is you see the hole, the one with holes. That's the cover. That's like the membrane that cover the nucleus. The plasma membrane is also another envelope. Each organelle, the mitochondria, your Golgi body, all of those have holes in them. Okay, so to regulate what enters, they are covered. They are all covered by a membrane. They are all covered by a membrane. Okay? Yeah. What's the question?
Let me show you. Yeah, what is here? Yeah, not the outside, right? The inside. Yeah, right there, yeah.
This one? So you have the nuclear envelope inside. I'm sorry, outside. You didn't have the nuclear envelope outside. Then that is your nucleus. And the nucleus has the DNA, the chromatin. And then inside you have your nucleolus. Yeah. I don't understand what you guys pointing to. No, but that one, that's your job. That one I can't tell you the answer to the exam question. You have to, I already told you what is it now. That's your job now.
The little purple ball is the nucleus.
No, the little purple ball is the nucleolus. See, this one is the ball is the nucleolus. Okay. Then above it, that's your DNA. Then above that is your envelope. But you will see your choices. You will see the answer choices. So prepare yourself. To study everything you'll be. So they're gonna give you answers like this here is the mitochondria. Does that make sense?
You give it an example.
Yeah. So they're gonna tell you this is the mitochondria, is it? So you're gonna be able to rule it out. So don't worry. Don't worry. If you study the cell, you know the structures, the answer is gonna be easy. So we're not playing games and tricks. I'm showing you exactly how like my friend said, I lifted it and I put it back to you here for you guys. Okay, so somebody said, when I finish my study guide, can I send it to you and you look at it and tell me if I got something wrong? No. You will proofread for you your work. I'm looking for the wrong ones, so I take point off. You want me to say, no, go fix this one. No, that's not how it works. You want me to proofread this for like 700 students? Unbelievable. No, it's impossible. But it's easy. It's not hard. I'm not trying to challenge you. I'm trying to guide you to the A. So everything is there. So I can give you, listen, this is too much work even for me. I make study guides for 700 students. I could make, listen, the question is 20 questions for study. What you get right, you go on, you get wrong, you fail. But I don't want that. I want to secure, give you the confidence. So you go in the exam, you know what you're dealing with, you finish I've been doing this. All my students, last semester, everybody got A, two students got B plus, and even one of them failed one of the exams. So be very, but maybe your school policy may say no. But you guys, please go study. There's no tricks here. Everything is laid out for you. That's in here. You see that graph is from there. That's the same one. You just study the organelles, study the structures. I'm not even supposed to put like the A, B, C, D. So now you only need to know those. But I put it. Okay, maybe only for the first exam. But in the future, you need to intensify some more. Of course, yeah. Yeah. You are supposed to know that's your cell. And they're not that hard, by the way. You look a lot, it's not that hard as the envelope, there's the nuclear envelope, there's the mitochondria, and the hemidesmosome, they connect the cells together. We're gonna go into each organelle by itself. Okay? Okay, now we're going to begin with region 1. Now we begin from very far outside, from the continent now. Are we in South America? Are we in Africa? Are we in Europe? Are we are we in North America? So if you want to study the cell, we begin with the surface. We already alluded to that today when we drew the cell membrane and the embedded proteins. The plasma membrane is so important because if you don't understand it well, when we move on to the nervous system and we go through the action potential, then you will not remember. So the plasma membrane defines the boundaries of the cell. And it appears as a pair of dark parallel lines when viewed with the electron microscope. They appear as a pair of parallel lines when viewed with the electron microscope. It has intracellular phase and extracellular phase. So the membrane is made up of lipid. Is 98% of your membrane is like butter. But remember, those lipids, they are not facing water because our body tissues are bathing in water. So how is that possible? How could our membrane lipid be dissolving in water? Because that particular type of lipid has a phospholipid, has a phosphate group on the surface, that allows it to bind with water. And that's the phospholipid. Please remember that. This phospholipid is 75% of the membrane lipids. So there's amphipathic molecules arranged in a bilayer. This is an amphipathic molecule arranged in a bilayer. So there's hydrophobic phosphate heads that face the water on each side of the membrane. And there's hydrophobic tails that are directed toward the center, avoiding water, and they drift laterally and they keep the membrane fluid. The cholesterol, 20% of the cholesterol is 20% of the membrane lipids, and it holds phospholipids still and can stiffen the membrane. Please remember that. The cholesterol causes stiffness, the cholesterol makes the cell membrane very tight, very strong. So the membrane lipids, membrane lipids are glycolipids. They are glycolipids, and 5%. So the membrane lipids, these are known as glycolipids, and 5% of the membrane, they represent about 5% of the membrane lipids. What are glycolipids? Glycolipids are lipids that are associated with sugars. And then there's glycoproteins. Glycoproteins are proteins associated with sugars, glycoproteins. And they contribute to your glycocalyx. It's a carbohydrate coating of the cell surface. Okay, guys, This is one of the most important figures you should know, and this is one of the most important figures for the entire course. So what do we have here? You see this yellow, all of this yellow, that represents your phospholipid. You see the head is circled. You see all these circles? That's the phosphate part. That's the phosphate part of that's why they call it phospholipid. It's not just a lipid, it's a phospholipid. So what's the difference between just being a lipid and being a phospholipid? This was on the other slide. I wish I can add that slide to here. Yeah, I can. Very good. Let me add it to this lecture. Right here. Yeah. This phospholipid. So a lipid is, oh, very nice. Okay, so do you guys see what a lipid looks like? It has, all of these are hydrophobic. Do you see any polar group? There's no polar group. It's all hydrogen and it's all carbon, 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 carbon. Here, now that's a phospholipid. That's not just a lipid. So what makes that's you see our glycerol? You see the green? You see glycerol? That's glycerol. You see where my mouse is? That's the glycerol. You see the glycerol, guys? Please look here, guys. Please. That's the glycerol. You see the glycerol? Let me get you the other picture. Okay, that is the glycerol. Do you see the glycerol? It has how many carbon? 1, 2, 3. That's the glycerol. When you attach them together, they look like that. That's the glycerol with long chain of fatty acid, carbon, long chain of fatty acids, carbon, long chain of fatty acids. Then you see the glycerol? That's the first carbon, second carbon, third carbon. It has long chain of fatty acid. The one in the middle got 12. It's got 12 long chain of fatty acid. But look at the first one. Look at the first one, guys. The first one, does it have a long chain of fatty acid like here? So that's glycerol. But a phospholipid, the glycerol has a long chain of fatty acid to carbon 1, long chain of fatty acid to carbon 2, long chain of fatty acid to carbon 3. But here, you got long chain of fatty acid to carbon 1, long chain of fatty acid to carbon 2. Now look at carbon 3. What does it have? Does it have a long chain of fatty acids? What does it have? Phosphate. Phosphate and what else? Choline. That's not a sugar. That is not a, who said sugar? Does this look like glucose? We forgot what sugars look like already. We forgot what sugars look like already. Does it look like that? So why does somebody would say this look like a sugar? This is not a sugar. Is this an amino acid? No, because you don't have an alpha carbon with 4 terminals. That's called a nitrogen containing choline group. But the base, what's the base? That's your glycerol. So we still a fat. Is that still a fat or no? Still a fat because you have a glycerol with a long chain of fatty acid like here. It's like you're looking at this, but only one of them is replaced with what? With phosphate. Thank you. With phosphate, is it just phosphate and choline? So that phosphate is what make it look like a polar head. So now that circle, that's the phosphate. And the other two tails are these guys. The other two tails are these two. These are the tails. And that's the phosphate, that's the head. That's the head, tails, tails. So now we have 2 in 1. It's like you have a car and an airplane. You have both. So you have the tail as carbon, hydrophobic lipid, and the head, they are polar. They are polar. So they can bathe with water on this side, but the lipid, they are in the core. And these guys with water on the inside and lipid in the core. Remember what I was saying earlier? Cholesterol can penetrate through so easily. Can penetrate through so easily. So easily. Can penetrate through. Because it's made of lipid too. Aldosterone, testosterone. Aldosterone, testosterone, and all of these all of these different lipid based steroid hormones, they can penetrate through. Okay, you see? But if you have something polar, can it enter? If you have, it cannot, because you cannot have something polar enter through the butter. This is butter. This is hydrophobic. Only the lipid cholesterol can enter, but water cannot. That's why you need a tunnel, a channel to allow water to go in. What about sodium? Can sodium go through this butter, through this thick fat tissue? It cannot, because sodium is an ion, is positively charged. It cannot dissolve in a lipid. What about glucose? Can glucose go through this thick lipid fat layer? It cannot, because glucose is polar or non-polar. Look at glucose. Is glucose polar or non-polar? It's polar. So how could polar go through this non-polar? It cannot. That's why you need a channel for glucose. You need a channel for sodium. You need a channel for potassium. You need a channel for water. So you need channel for all the stuff that cannot go through the lipid bilayer. Is that point clear? That point is very important. So now you see that in the membrane, you have two leaflets, one on the top, phospholipid bilayer, an upper leaflet with phospholipid head and hydrophobic tails, with a phospho head and tails of lipids. And you have some proteins that span both. They cross both. They are like a tunnel. These are called transmembrane, because they are traversing both layers of the membrane. That's transmembrane. But if it's only on one side, that is known as, peripheral proteins. These are peripheral because they are on the periphery. So not all the proteins span both layers, 1 and 2. Some of them, they stuck on one, they are on the peripheral, the upper side, and some of them are stuck only to the bottom. So these are known as peripheral proteins. Peripheral proteins do not span both layers. They are stuck only to one in the bottom or one at the top. It's so important for you to know that. Can somebody please tell me the difference between a transmembrane and a peripheral membrane? Yeah, excellent. Excellent. So the transmembrane, what then you also responsible of knowing, give me examples for transmembrane protein. Who are those transmembrane proteins? Example, a perfect example of that would be who? A channel, water channel, sodium channel, ion channel, to allow the passage to the inside. What about only on one side? These peripheral proteins, that's an example of axillary proteins. So the one on the top, they could be a part of the receptor membrane that binds the ligand. Remember binding the ligand? Okay, what are glycoproteins? So now we have glycolipid. So green, what does green represent? Green represents glucose. So we have glycolipids and glycoproteins. Remember, glycolipids and glycoproteins. You remember, guys? Glycolipids and glycoproteins. And these are carbohydrate chains. So now Glycolipid, that means that sugar attached to the phospholipid bilayer. And glycoprotein is sugar attached to the protein, the transmembrane protein or peripheral proteins. From the name you can tell, glycolipid is, glyco is sugar. Glycolipid, sugar attached to the fat region. Glycoprotein is sugar attached to the protein. Then membrane proteins. Membrane proteins constitute 2% of the molecule, but 50% of the weight of the membrane. Wow, they are only 2%, but they are, that's like you have 100 people and 2 of them weigh about 50% of the weight of everybody on the scale. That shows you how big these transmembrane proteins are. And we have 2 different types of proteins. There's transmembrane proteins. These pass completely through the membrane with a hydrophilic region that contact the watery cytoplasm and extracellular hydrophobic regions that pass through the lipid of the membrane. So most are glycoproteins. Most are glycoproteins. So some drift in the membrane and others are anchored to the cytoskeleton. Others are anchored to the cytoskeleton. So peripheral proteins adhere to one face of the membrane. Peripheral proteins adhere to one face of the membrane. So those on the interface are usually tethered to a transmembrane protein and the cytoskeleton. Okay? So, transmembrane proteins. Transmembrane proteins span. That's so important to know. You see the phospholipid bilayer? They span both regions. While here, you have also they span both regions. But the transmembrane is just like the plasma membrane. It has a hydrophobic core, and it also has, because that's water up here, there's water down here, so the transmembrane also must have a hydrophobic core with the butter to dissolve here, and a hydrophilic head to dissolve in the water region. Okay? That's a transmembrane. Now, examples of membrane proteins. They could be receptors that bind chemical signals to trigger internal changes, and they may cause production of a second messenger within the cell. We're going to talk about the second messenger. Also, we said enzymes that catalyze the reactions, including the digestion of the molecule and production of second messengers So the channel proteins. Channel proteins allow hydrophilic solutes and water to pass through the membrane. So some are always open, called leak channels, and some are gates, known as gated channels. Okay, let's talk about that. Yeah, this is really, really important. Okay, now when we are talking about, so now we will remember, we always go from big to small. We already identified the cell membrane, and the cell membrane has different types of proteins that span both layers. So now let us draw the plasma membrane. So now we're going to draw the plasma membrane. So these are the heads, and it has 2 layers. So it has an upper layer and it has a lower layer. So both of them, they have phospholipid heads, and they also have tails. They have heads and they have tails. They have heads and they have tails. They have heads and they have tails. So the heads are polar because they have phosphate, and the tails are non-polar. So we already said, Mr. Cholesterol, Cholesterol, some hormones are structurally based off of cholesterol, such as testosterone, aldosterone, progesterone, estrogen. These guys are made up of cholesterol. So when they're out here, they can go through so smoothly because the bottom core here is hydrophobic and lipid, just like them. It's cholesterol too. So they can cross so easily, and they bind receptors in the cytoplasm, and there are receptors also inside the nucleus in the DNA, and then they bind them, activating them, causing transcription. So then we can have central dogma of biology. We can have transcription, then translation, then protein. So you see, the point is they cross so easy. That's known as the first messenger. So here the first messenger can cross, bind here, give me a target effect, or cross, bind here, give me a target effect. But now let's look at the problem of being polar. If you are polar, that means you cannot cross. So if your first messenger is polar, if the first messenger is non-polar, which is the same thing as hydrophobic, non-polar, hydrophobic, it can cross so easily. But it's hydrophobic. But if it's hydrophilic, can it cross? So we need a target effect, right? We need a target effect. So here the first messenger gave him a target effect. So the first messenger did everything. It came, passed through, stimulated the receptor, give me a target effect. What about the first messenger here? It can never enter. So what are we supposed to do now? So I need a channel. That's a good guess. But that's not what's going to happen. I need a channel for nutrient, for glucose. I need a channel for electrolytes, sodium and potassium. But for a hormone to do its job, then I don't need a channel because the system has solved it differently. You're going to have a receptor. A receptor is a structure that is, do you think the receptor is peripheral or transmembrane? The part of it that binds my first messenger, right, it's peripheral. But I didn't ask about the receptor. I asked about the whole receptor. The whole receptor is definitely transmembrane. And it has another peripheral receptor. It has another peripheral protein down here. It has two peripheral proteins. One to bind my first messenger, and when it binds the first messenger, it's gonna make conformational change. This one here, the peripheral receptor here, is gonna know, oh, peripheral receptor number one must have bound the ligand. And the ligand is the first messenger who cannot enter. Don't worry, you cannot enter, I'm here. Then this one is going to go and activate for me an enzyme here known as, on the cell membrane also, is the transmembrane enzyme known as adenylate cyclase. And adenylate cyclase now is going to convert for me ATP, adenine triphosphate, into AMP, adenine monophosphate, then adenine monophosphate into cyclic, because remember it's called adenyl cyclase, so it's cyclic. So it's going to convert it into a cyclic AMP. Cyclic AMP is going to turn from off to on an enzyme known as protein kinase. And the protein kinase is going to phosphorylate for me my target enzyme who will do my job. The target enzyme was off, but the kinase is going to turn it into on, and then when it's on, it's going to do my target effect. So what I need you to know is that the first messenger couldn't get in, but a second messenger, this one, the cyclic AMP, that's the second messenger who went to resolve everything and give me my target effect. So yeah. So what are we saying here? If you are a first messenger who is non-polar, hydrophobic, steroid-based, cholesterol-based, you can cross so easily. But if you are if you are polar, you can never cross. If you are polar, you can never cross. You only bind on the outside, and a dimerization happen, and that will cause the initiation of the signal to go and to convert for you, activate a second messenger. Then the second messenger will give you a target effect. So I needed target effect in both cases. But here I achieved my target effect by myself as the first messenger. But there I needed a second messenger. Do you understand? Why do I need a second messenger? Somebody come outside, they have ID, they go in, they do what they want, come out. But somebody who doesn't have an ID, they stop. Somebody on their behalf goes in because they have the ID to be inside, and then they can do it for them. Same thing in the cell. Okay, so these are receptors. We have proteins who are enzymes, and we talked about the channel. So this channel that I just showed you, this channel, we have channels that open for different reasons. So if I have a channel, what kind of channels do I have? The first one is called leak channel. Leak channel. That's like the gate in the park. That's open for everybody. The gate in the park is open for everybody. Can enter and exit. But these are called leak channels. So leak channels allow electrolytes to flow in and out freely. However, there are ligand-gated channels. What do you think will happen with ligand-gated channels? What happens with ligand-gated channels? In ligand-gated channels, they are closed, but when the right ligand, acetylcholine, binds, it will open. If the ligand leaves, it will close. So here, who causes the opening is the ligand. So the ligand causes the opening. That's a ligand-gated channel. Or sometimes you can have a channel right here. This channel is not opened by a ligand. It is opened by membrane potential changes, voltage. They're called voltage dependent or voltage gated channels. They open to allow the influx of their ions. So how many kinds of channels I have? I have leak channels. They're open like the park. They're always open. Everybody can go through them in and out. And then I have the gated ones. These are gated. So the gated ones can only open in 3 situations. Either with a what? To open. The gated channels. What are the things that make it open? Yes?
Oh, the A word up there.
Ligand?
The ligand,
The ligand makes it open. Like acetylcholine. That's an example of a
That's the
ligand.
one I was talking about.
The second one is the voltage. If the membrane electricity changes, causing it to open. And the third one is mechanoreceptor. So that means when somebody touches you, why do you feel it? Because the channel is open, the electrochemical entered, and then you have a particular type of ion flowing in. Okay? Okay. Give me an example of the reason why would a gated channel that's closed open? What are the things that can make it open? Give me one of them. Chemicals. What do you mean by chemicals? Like what will lead, like it's closed, so somebody got to open it. What are the things that make it open? We just mentioned 3. So give me one thing that makes the channel open. Yeah, so chemical messengers or ligands binding to a ligand, and that causes the gate to open. Give me a second reason why would a gated channel open besides binding to a ligand. Mechanical, excellent. So if you touch it, these are known as mechanoreceptors. These are the ones who get activated in childbirth. When the baby is pushing against the cervix, then they get stimulated, they get open, they send a message, electrical message to the brain. We will talk about that. Also, another example is when the baby ejecting the milk. The baby suckling the breast milk, stimulated in the nipple, then all the milk duct is going to come out of the milk is going to come out of the milk duct. What's the third one? Voltage gain. So when electricity changes, the action protection, the electricity changes, excellent. That's going to cause the very good, very good, very good. Very good, 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 very good. Okay. 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 Membrane proteins. Membrane proteins. So the functions of membrane proteins. These are carriers that bind the solutes and transfer them across the membrane. These are carriers that bind the solutes and transfer them across the membrane. Carriers that bind the solutes and transfer them across the membrane. So these are the membrane proteins, the transmembrane proteins. These are pumps that consume ATP. There are cell identity markers, and these are glycoproteins acting as identification tags. And there are cell adhesion molecules that mechanically link the cells together. This is so important. That shows you all the different types of cells, all the different types of the cells, and of the transmembranes that are found on the cell. It could either be a receptor, and a receptor binds this red triangle ligand, and a receptor that binds to the chemical messenger, such as hormones sent by other cells. Then there's an enzyme. So an enzyme that breaks down a chemical messenger and terminates its effect. An enzyme that breaks down a chemical messenger and terminates its effect. Channel is a channel protein that is constantly open and allows solutes to pass into and out of the cell. So a channel protein that is constantly open and allows solutes to pass into and out of the cell. So that is a leak channel. What kind of channel is that? Is a leak. Does it need a ligand? Does it need a voltage? It doesn't. Now look at this one. Now that's gated. So that's a gate channel that opens and closes to allow the solutes through only at certain times. Only at certain times. There are also cell identity markers. Cell identity markers. So cell identity markers are a glycoprotein acting as a cell identity marker, distinguishing the body's own cells from foreign cells. And there are cell adhesion molecules, a cell adhesion molecule that binds one cell to another. Okay, second messengers. An example of a second messenger system is stimulation of a cell by epinephrine. So now we have already talked about that. So the first messenger here is epinephrine, bind to a surface receptor. That immediately tells you that epinephrine is polar or non-polar. If it binds a surface receptor, it cannot enter. It binds a surface receptor. That makes it Polar or non-polar? Polar, excellent. The receptor activates a peripheral protein. You see this little guy, this little brown guy that goes, you see this one, the brown one. That's the G protein that's gonna go and say, oh, somebody outside, he wants something to do. So now he's gonna go mediate. So the G protein is an intercellular peripheral protein that gets energy from guanosine triphosphate, GTP. So the G protein relays a signal to adenylate cyclase, which converts ATP to cyclic AMP, the second messenger. Cyclic AMP activates the cytoplasmic kinases. And the kinases add a phosphate group to other enzymes, turning some on and others off. Up to 60% of drugs work through this G protein and second messenger. Right here, perfect. Look at this. I told you, anytime you see a photo like that, it's either going to be on the exam itself is going to be on the exam, or the concept that is trying to tell you it's going to be on the exam. So look here, a messenger such as epinephrine, red triangle, binds to a receptor in the plasma membrane. It binds to a receptor in the plasma membrane. So the first messenger is a receptor, and the receptor releases a G protein. You see the G protein, which then travels freely in the cytoplasm and can go on to step 3 or have various other effects on the cell. So we're going to convert GTP to cyclic AMP. And the G protein binds to an enzyme, adenylate cyclase, in the plasma membrane. Adenylate cyclase converts ATP to cyclic AMP. And then inactive kinase will be activated and inactive enzymes will be activated. Okay. The glycocalyx. The glycocalyx is a carbohydrate moiety of glycoproteins and glycolipids external to the plasma membrane. Is unique to everyone. Unique to everyone, but identical twins. This glycocalyx is really important, guys. This glycocalyx is so important. The glycocalyx is a set of sugars that are at the top of the membrane. How many kinds of sugars? We already mentioned them in this slide. We already mentioned them. How many sugars do we have? Sugars with lipids, glycolipids, or sugars with proteins, glycoproteins. So these are the two types of sugars on the surface. Your glycolipid, sugars attached to lipids, and glycoproteins, sugars attached to proteins. Sugars attached to proteins. Very important. So now the sugars attached to the lipids on your body and the sugars attached to the protein on your body, are they the same that everybody else's? No, these are known as the glycocalyx. That's like your social security number. So the sugars on, how many sugars do we say we have on the surface on our skin? 2. What are they? Glycolipids and glycoproteins. So carbohydrate moieties of glycoproteins and glycolipids external to the plasma membrane. So unique in everyone but identical twins. The functions are protection, immunity to infection, defense against cancer, transplant compatibility, cell adhesion, fertilization, embryonic development. So microvilli are extensions of the membrane that serve to increase the surface area. So the microvilli are extensions of the membrane that serve to increase the surface area. They provide 15 to 40 times more surface area to cells that have them. So best developed in cells specialized in absorption. On some absorptive cells, they are very dense and appear as fringe, and they're called the brush border. So this is a type of an extension. So we finished now with the glycocalyx. We're now going to talk about the extensions. I already alluded to them. We have 4 extensions you guys need to know. Number 1, the microvilli. Number 2, the cilia. Number 3, the flagella. Number 4, the pseudopods. These are the extensions of your cell membrane that you got to know. Let's begin with the first one, microvilli. The microvilli are on your intestine. And the intestine, there are so many on the intestine because they increase the surface area. This is so important because you could be asked, what is the usefulness? What is the usefulness of microvilli? It's to increase the surface area. You could be asked, provide 15, they could be asked, where exactly are they? They are in the absorptive brush border. So on some absorptive cells, they are very dense and appear as fringe and they are called the brush border. So some microvilli contain actin filaments that are tugged toward the center of the cell to milk the absorbed content. So remember, microvilli, surface area, microvilli, surface area, brush border. Microvilli, surface area, brush border. The 4th thing is they are made up of microfilaments. Microfilaments. So these 4 things go together. These 4 things go together. The microvilli increase the surface area. They are made up of microfilaments and they are found on the brush border of your intestine. That's them. So when they increase the surface area, they're going to increase the rate of diffusion. Diffusion. Excellent. Now let's move on to cilia. That's the second type. We finished with the first type. Remember we said we have 4, microvilli, the cilia, the pseudopods, and the flagella. So that's number 2. That's the cilia. And the cilia have axonemes. And the cilia, they are not there for the surface area. They are there for movement. So they shove, they push things. So they shove and push things. That's why they are found in your respiratory tract to send down mucus. To send down mucus and to send down all of to send down mucus and to send down the material that needs to be flushed. So what's the key word? Cilia. And they have an axoneme. They have an axoneme. The cilia have an axoneme. And they are made up of microtubules. The cilia have axonemes. And they are made up of not microfilament. They are not made up of microfilament. What are they made up of? Microtubules. And they have dining arms and they are there for movement. This is all we need to know. Cilia, they have an axoneme. Where do we find the axoneme? Do we find it on the microfilament or we find it on the cilia? And the microfilament are there for absorption, a brush border, and they're there to increase the surface area. But the cilia, they have nothing to do with surface area or brush border. They are there for movement. You see, and they move with something known as the power stroke and the recovery stroke. We're going to see that in a little bit. The power stroke followed by the recovery stroke. Power stroke followed by a recovery stroke. You see the cilia? And this is the axoneme. The axoneme looks like that. It has dining arms, and these are two central macrotubules surrounded by 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's the axoneme. You see the ciliary action? They shove down and move the mucus. You see the power stroke? the powder stroke, they move things down. They shove down mucus in a powder stroke, and they recover, stand up again. That's recovery. So they go like that. They shove down with a power stroke, then they rise again with a recovery stroke. That's the cilia. That's not the microfilament. So now cystic fibrosis is a disease where it's a hereditary disease where you can have chloride channels, but you can never plug them into the membrane. So there will always be chloride on the surface. Flagellum. Flagellum is a whip-like structure. Is just like the cilia, so that means it will also have an axoneme, and it's much longer than the cilia. They are whip-like structures, so that's what the sperm is all about. That's the sperm. It's flagellum, whip-like structure, and it has an axoneme, you see, identical to the cilium. That's the perfect question. Who is identical to the cilium? Who is identical to the flagellum? Is it the cilia or the microfilament? The cilia, because it has an axoneme identical to the cilium. And they move in an undulating snake-like corkscrew. Okay, pseudopods. Pseudopods are extensions of the cell surface. Continually changing extensions of the cell that vary in shape and size, and they could be used for cellular locomotion, capturing foreign particles. So that's the pseudopods. Who uses pseudopods? That's like the macrophage. Remember the macrophage? They're like extensions and projections that make them crawl. These are the pseudopods who crawl. And that's for locomotion and capturing foreign particles. That sounds like the macrophage who keeps moving from one place to another to look for bacteria and fungi and infectious microbes to swallow and engulf them. Okay, so now we're gonna move into the membrane transport. Now we looked at what happens in the membrane. Now we're gonna look at the transport processes. All of this is just extra. Let me get that out the way. Okay, membrane transport. So the plasma membrane. Okay, so we know that the plasma membrane and organelle membranes are selectively permeable, allowing some things through but preventing others from passing. So the passive mechanisms require no ATP. If you're moving passively, you go from an area of a higher concentration gradient to an area of a lower concentration gradient. And that is a perfect example of passive mechanisms would be filtration, diffusion, and osmosis. We use no ATP. Does that mean we're not using any other form of energy? No, we're just not using ATP. But in simple diffusion, we use kinetic energy. That's a different type of movement. But we only use ATP in active transport. And as we mentioned, just because there is a lot of glucose outside than inside, can glucose flow through the lipid bilayer? No. So it needs a pore to help it, a carrier to grab it from outside and get it through the inside. That is known as a facilitated carrier-mediated diffusion. So now this is filtration. That's how your heart, when your heart pump, it pump with so much pressure, the pressure of the blood now starts to move away from the area of the pressure to the capillary that is fenestrated with holes, and then all of the material starts to come out. The filtration through the wall of the blood capillary. So the solutes are moving out of the blood due to the action of the heart. That's known as hydrostatic pressure. So hydrostatic pressure is when the heart is pumping the blood. The heart is pumping the blood and the blood is leaving the capillary. The blood is leaving the capillaries and exiting. You see these arrows? So now all of these particles are leaving out from this blood vessel into the surrounding area. This is known as when you are exiting through the clefts. That's like you're filtering. That's filtration. You see, they are filtering, and who's powering the movement of the filtration? It is the hydrostatic pressure from your heart. So the heart is pumping blood, and the blood is running through the capillaries, and the pressure is so much leading these guys to diffuse out. Simple diffusion means when we flow from an area of a higher concentration to an area of a lower concentration. So the net movement of particles from place of high concentration to a place of lower concentration due to the constant spontaneous molecular motion. So molecules collide and bounce off of each other, and substances diffuse down their concentration gradient. So from an area of a higher concentration to an area of a lower concentration. And that occurs in the air, and water does not require a membrane. So substances can diffuse through a membrane if the membrane is permeable to the substance. Okay, so the factors affecting diffusion. So what can lead to a faster diffusion rate, and what could cause a slower diffusion rate? First of all, the temperature. So when you increase the temperature, you increase the rate of motion of the particles, you increase the rate of motion of the particles, and then the molecular weight would increase, and larger molecules would move slower. So steepness, there would be steepness of the concentration gradient, and that would increase the difference and the rate of transfer. So membrane surface areas. In the membrane surface area, there's an increase in the surface area, increasing the rate of diffusion. In the steepness, you would have an increase in the difference between the two areas, leading to an increase in the rate of diffusion. And there are larger molecules that move slower. So, and when the membrane permeability increases, that increases the rate of diffusion. So these are the factors that allow for an increased rate in the diffusion. So osmosis, water has very important substance and properties for life. We said that water has high specific heat, high thermal stability. Water has high cohesion, and water has very high adhesion properties. And in addition, water is important for hydrolysis to separate macromolecules. Water is important for dehydration, removing water molecules to join together the macromolecules. So in osmosis, if you have a membrane and the membrane is only permeable to water, what's going to happen is water is going to move from an area of less solute, not less water concentration, water of less solute to an area of high solute, because water is a versatile solvent, will go to dissolve all of these solvents. So osmotic pressure. Osmotic pressure represents, osmotic pressure for me represents the incentive to go and dilute from an area of a lower solute pressure to an area of a higher solute pressure. So that increases as the amount of non-permeating solutes rise. The hydrostatic pressure is the fluid pressure on the membrane. That's the fluid pressure on the membrane. There's also reverse osmosis, where the process of applying mechanical pressure to override the osmotic pressure, and that allows the purification of water. So we must have an excellent understanding of osmolarity and tonicity. So osmotic concentration is the quantity of non-permeating solutes per liter of a solute. So the body fluids contain a mix of many chemicals, and the osmolarity is the total osmotic concentration of all the solutes. So the blood plasma, the tissue fluid, and intercellular fluid are 300 milliosmoles per liter. I'm gonna wait for your friend to get back so I can do the illustration on the board, because now we have to understand the difference between tonicity and osmolarity. That's the tonicity is the ability of a surrounding solution to affect the fluid volume and pressure in the cell. And that depends on the concentration of non-permeating solutes. So if a solute is more concentrated on one side of the membrane, it causes osmosis. Okay, tonicity. So we can have a hypotonic solution. A hypotonic solution is a solution that has less osmolarity than will tend to absorb water. It's a hypotonic solution, causes the cell to absorb water. So if we're describing a hypotonic solution versus a hypertonic solution, we are describing what would this solution do to water. Would it absorb water, or would it expel water? Or are we going to have an isotonic solution? So we must understand the three scenarios. We must understand what could lead to a hypotonic solution. What are the effects of a hypotonic solution on a red blood cell? What are the effects of hypertonic solution on red blood cell? And what are the effects of an isotonic solution on a red blood cell? What are the effects on a red blood cell? What are the effects on the red blood cell? So now look at this blood cell. What happened to it? It swelled. So it increased in size. That's the normal size, and now it swelled. And this is a cell that is at the same size. That means it's in a isotonic solution. And that's a cell that shrink. So this cell that shrink, it has lost the solution. The water has exited from the cell into the solution. So this unit here was teaching us about the importance of being selectively permeable. We already discussed that this plasma membrane is able to exclude some substance and allow the passage of some other substances. So what can the plasma membrane allow the entry of? The plasma membrane allows the entry of lipophilic substances that will easily dissolve in lipids. These are the ones that will be able to penetrate and enter the cell membrane. But hydrophobic and lipophilic can enter easily. Hydrophilic and lipophobic can be expelled. So in filtration, particles are driven through the membrane by physical pressure. And we said, what's an example of filtration pressure? That is when the filtered water and small molecules pass through the gaps between the cells or the filtration pores of the capillary wall. That is an example of hydrostatic pressure powering the movement of these solutes through powering the movement of the solutes through the through powering the movement of the solutes through the blood vessels. And that allows the delivery of water and nutrients to the tissues. And it allows the removal of waste from capillaries and the kidneys. Okay, filtration through the wall of a blood capillary. So the blood pressure in a capillary forces the water and the small solutes such as salt through the narrow clefts between the capillary cells. So you see now we are diffusing. So all the substance is exiting, all the substance is leaving through the pores because who's powering the movement? You will never have movement unless there is some energy powering it. Sometimes you have chemical energy, ATP, causing the movement. And sometimes you have solutes that are moving not because of ATP, but because of hydrostatic pressure. So here the hydrostatic pressure is causing, for the movement of hydrostatic pressure causing the movement of the solutes through these clefts. So simple diffusion is where there is a net movement of particles from a place of a high concentration to a place of a lower concentration. And that is due to constant spontaneous molecular motion, and the molecules collide and bounce off each other. So the substances diffuse down their concentration gradient from an area of a high concentration to an area of a low concentration. So in order for diffusion to happen, you must remember these factors, guys. What causes the rate of diffusion to be faster? What causes it to be slower? Temperature. What is the effect of temperature on the rate of diffusion? The more temperature, the faster the molecules are going to move. Molecular weight. The smaller you are, the faster you're going to move. Steepness of the concentration. The bigger the difference between the two areas, the faster the diffusion rate will be. And there's membrane surface area. So the higher the surface area, the higher the rate. And there's membrane permeability. The higher the membrane permeability, the higher the rate. So we know that there are aquaporins. We said that water is polar and water cannot penetrate the membrane. So it needs protein channels to allow the water to enter. And these are known as aquaporins. So in osmosis, the water enters from side A into side B, and water moved. So why didn't the solute move from here to the other side? Why did not the solute move from this side to side B? Why didn't the solute move from side B into side A? Why only from side A to side B? So because here the bilayer is only allowing the passage of water. The solutes cannot. So here water moved from this area to this area, and that increased the water volume. That increased because water has a tendency to go and dissolve. So solute cannot move to this side. The pores are too small. So water will go to diffuse this side. So now you will see that water is looking like that, diffusion. So that's a hypertonic solution. And in order to move, we don't just need a gradient. We need a gradient, and we need to provide carriers to allow for the passage of these molecules. So carriers are proteins in the cell membrane that carry the solutes into and out of the cell. The carriers exhibit specificity for their particular solutes. And the solute ligand binds to the receptor site on the carrier protein, and the solute is released unchanged on other side of the membrane. So carriers also exhibit saturation. So as the solute concentration rises, the rate of transport rises as well, but only to a point that is called the transport maximum. Okay, guys. I'm waiting for everybody to get back so we can do the illustration and nobody misses it. This is very, very important. We know that already in order for movement to happen, we need some form of energy to power it. So we have already established in the past that there is a diffusion gradient. We said that if you have two sides where there is a selectively permeable membrane only allowing, like let's say we begin with water. You have 600 milliliters of water, 100 milliliters of water, then all the water is going to go from 600. Let's say you have 500 here and 100 here, then the water is going to go from an area of a high concentration to an area of a lower concentration till they both equilibrate at 300. If you replace it with temperature, 500 degrees Celsius, 100 degrees Celsius, and you put, they are a slab of iron, you make them touch, then the temperature is going to even out. And you're going to have 300 on one side, 300 on the other side. They're going to look like that. 300 degrees Celsius on one side, 300 degrees Celsius on the other side. If you had sodium, 500 millimoles of sodium plus and 100 millimoles of sodium plus, and here you have 500 chloride minus, and here you had 100 chloride minus, they will even out at an electrochemical gradient of 300 sodium chloride on one side, 300 sodium chloride on one side. So that represents for you the diffusion from one area to the other. Then we talked about a special type of diffusion, and that's water. That is known as osmosis. So water moves from an area of a higher concentration of water to an area of lower concentration of water. But let's say you're not looking at that no more. You're looking, you have a beaker like that, and you have 1 liter of water here and 1 liter of water here. So the water between side A and side B is the same. The water is not the difference. The difference is you make this membrane only permeable to water, and you put on this side 500 millimoles of sodium chloride, and here you put 100 millimoles of sodium chloride. What's going to happen now? Don't tell me sodium going to go from 500 to 100, because I already said that this line only allows the passage of water, because water is so small. H2O, O with two H's, much smaller than NaCl. These are so big, they cannot cross. Only water can cross. What can happen now? So the water, the 100?
Not the 100, the water from the 100 is
is going to go from an area of a lower solute concentration to an area of a higher solute concentration. Are we clear? So that movement, did it require ATP? What is ATP a kind of? A kind of what kind of energy? That's a chemical energy. That is chemical energy. Do we only have chemical energy in life? No, there's other types of energy. So these guys moving, they're not requiring ATP, but they obviously have some other, that you will never move unless there's some energy that's making you move. What's the energy that is making these guys move? Kinetic energy. That's the kinetic, because the kinetic, what's kinetic energy? What is kinetic energy? You see, you are picking up. Sometimes, this is, we have the conscious mind and the subconscious mind. So I'm saying things. I'm saying so many things, and your mind is like, am I understanding? You are. because it's storing in your subconscious mind. You see that she said the right answer, she didn't even know it was right. So it's a lot, but be patient. That's how you're gonna get the knowledge. Just be patient. What happens now? This is known as, all of this is known as diffusion through kinetic energy. So now I don't want you just to say it, I want you to understand it. What is kinetic energy? Kinetic energy is the energy of bumping. So there are so many, and now they are bumping to each other, so now they move in that direction. So if you put 500 millimoles of water over there, and 100 here, the ones bumping there is going to spread out until they even out. If you have a Petri dish, you put a drop of methylene blue here, and you put a drop of bromide on the other one, then they're gonna start, because they're so close together, they're gonna push, they're gonna have kinetic energy until they spread out. You get it? So that's the kind of an energy that we need. So don't think, students think, oh, secondary active transport requires energy, but diffusion does not require energy. That's not true. Diffusion does not require what kind of energy? Chemical energy in the form of ATP. Okay, then we move on to the tonicity. In tonicity, I can put my red blood cell that is supposed to look like discoid biconcave, depressed from the top and the bottom. Discoid biconcave, discoid biconcave, discoid biconcave. It looks like that. Discoid biconcave. If I put in a solution that is hyperosmotic, 700 milliosmoles, or I put it to it, the red blood cell has 300 milliosmoles. 300 milliosmoles. 300 milliosmoles. If I put it on beaker A, it has 700 milliosmoles. Beaker B has 300 milliosmoles. So obviously that's equal. That's too much. And here I put it in a 100 milliosmoles. So now that's two less. The beaker that has a lot of solution, very, very osmolar compared to the osmolarity of your red blood cell, that's known as hyperosmolar solution. That is isoosmolar solution. And that is hypoosmolar solution. So now what will the water do now? The water in the hyperosmolar solution is going to move from 300 to 700, like we said there, and then it will shrink. It will shrink because now the solution caused the cell to lose water. We call it hypertonic. And here the water will be even out. A little bit will enter, a little bit will exit. So they will be in a steady state. They will be in equilibrium. Okay? And that equilibrium is known as isotonic. And then the hypoosmolar, what's going to happen is the water is going to move from an area of a low concentration to the area of a high concentration. Is going to make it swell until it bursts. It's going to swell. When we call that, and then we call that solution hypotonic. That's very important. So we said one of the reasons why we move is kinetic energy, and there's also, that's for diffusion. How is diffusion different from filtration? In diffusion, the molecules are flowing based on kinetic energy of them colliding and bumping with each other. So many 500 here bumping to each other, spilling over, bumping and spilling over to each other. But in filtration, that's what happens in your heart. So in your heart, your heart looking like this. Your heart pumps blood to the tissue. And when it arrives to the tissue, there is a capillary full of holes. And then the capillary next to it is our tissue. Next to the capillary is our tissue. So what happens now, the solutes in the blood start to get out through the holes, through the pores. We call them fenestration. Fenestrations. So they get out through like that. Okay? So who's causing them to come out? It is the pressure of the heart. So when this heart compresses like a pump and push so hard, blood rushes out, it comes out like a toothpaste. So you're squeezing, that's called hydrostatic pressure. That's not kinetic, that's hydrostatic pressure. So it is the height, so filtration is based on hydrostatic pressure. That is so important, guys. Filtration is based on hydrostatic pressure. Diffusion, so when they ask you on the exam, what is filtration based on? Hydrostatic pressure. What does hydrostatic pressure power? Diffusion or filtration? What's the answer? Filtration. Okay, so now we're going to talk about carrier-mediated transport. So in the cell, so we talked about moving from an area of a high concentration to an area of a lower concentration. Moving from an area of a higher concentration to an area of a lower concentration. Now, in the membrane, in the plasma membrane, we have a different type of problem. These are the heads, and these are the heads of the lower leaflet. And these are the tails. Are the tails hydrophilic or hydrophobic? Are the tails hydrophilic or hydrophobic? Hydrophobic. And the heads, are they hydrophilic or hydrophobic? Okay, so now this is our cell membrane. So in this cell membrane, let's say I have a lot of glucose outside. And I have a lot of sodium outside. I have a lot of sodium outside, a lot of glucose outside. A little bit of sodium inside, and a little bit of glucose inside. So based on what we learned, what should be the direction of movement for sodium and glucose? What should be the direction of movement for sodium and glucose? If there is a lot of glucose outside, that's outside. And that's inside. What's going to be the direction of movement for these guys? That's influx. It's going to be an influx. They're supposed to go in the influx. But the only thing is, this is not this. It's not that. They can just pass through. That's a lipid bilayer. That's the hydrophobic core. Can glucose go through the hydrophobic core? It cannot. That's why it needs a membrane channel that will facilitate. It's a carrier. It's not just a channel. It will grab glucose. It has an arm. It will grab glucose, then shut off, then throw it out, then shut off, then open again. It's an actual carrier. Glucose carrier, glucose known as facilitator. So this is known as facilitated diffusion. So they are diffusing. They're still diffusion, but through a facilitator. through a carrier. That's why they call it facilitated diffusion. Sodium also. Sodium now, its movement down concentration gradient, because it's polar, has to be through a channel. It has to be through a channel. Goes through like that. Through a channel. Now, the movement of sodium, something very interesting. You know, sometimes somebody is already going from Manhattan to the Bronx. Somebody say, can I get a ride with you? And they ride. That's exactly what happens. You have a sodium, it's called the siglet. Siglet. What's a siglet? The siglet is a sodium glucose transporter. So when the sodium enter the cell, glucose will say, can I come with you? And glucose enter. Glucose enter. But there's a problem. Please pay attention. There's a big problem. When glucose enters the cell, sodium enters the cell because it's natural. Sodium goes in, glucose goes in. But glucose goes into glycolysis, as we said earlier, and glycolysis gives us pyruvate and gives us ATP. But the sodium keeps piling up, keep piling up, keep piling up. So now if sodium just goes in and stays here, it's gonna pile up. There's gonna be so much sodium, they're gonna have equilibrium, right? So what prevents sodium from piling up inside? You see, the cell is a circle, right? It doesn't only have one side. There is another side down here. There is another side down here. Known as the basolateral side. Is known as the basolateral side. So what happens here, there is a channel here known as the sodium potassium ATPase. This is really important. The sodium potassium ATPase, what it does is, when sodium is a lot here, it will take it from inside, throw it outside. But remember, outside, there's a lot of sodium. There's a lot of sodium already. So now what is the sodium potassium ATPase doing? It's taking sodium from where there is little, throwing it outside where there is a lot of sodium. Is that diffusion? That's against concentration gradient. Remember we talked about there is downhill gradient, that's down gradient, and then there is uphill when you go up. Then there is uphill gradient. The sodium potassium ATPase, what it does is it takes 3 sodium, Na, throwing it out against concentration from where there is little, throwing it a lot. And the opposite. Potassium is a lot inside and sodium is a little bit outside. It will take sodium from where there is a little bit, throwing it down by taking 2 potassium. So on the exam, there's a question. What is the sodium potassium ATPase? It's a membrane channel. It's an ATPase. So it uses ATP, convert ATP to ADP, and hydrolyze that energy to move sodium against concentration gradient from an area of a little concentration to an area of a lot. You know, remember Black Friday when the many people entering? Imagine somebody standing at the door, throwing all of them out and taking every little 3 people who walk in, throwing them out. That is exactly sodium potassium ATPase. It takes from where there is a little bit inside and throwing it against what's outside. So it takes 3 sodium from
gradient.
Huh?
Would it be considered like up gradient?
Yeah, that's against the concentration gradient, uphill. So it throws the 3 outside and takes inside, and takes, not, it's sodium, potassium, not just sodium. Takes 3 sodium, throwing them outside, and 2 potassium, throwing them inside. Throwing them inside. And that is what's gonna get rid of all these little sodiums. And when we get rid of the sodium, then the sodium can flow in on the other side, on the apical side. So it keeps depleting sodium from here so that we can have a flow of sodium here and glucose can enter so we can take it for ATP. We need this gradient because we need to shuttle the cingulate so it can carry glucose with it. So if we allow glucose goes in, we use it, it's gone. But the sodium keeps on piling up. So the sodium, potassium, ATPase take it on the basolateral side, throwing it outside. You will see it in a figure now. And remember that there is a transport maximum, and we see that a lot with diabetes. So patients who have diabetes, their urine has a lot of glucose. Why is that? Because the channels that take glucose from the urine, throwing them to the blood, are full. So now glucose start to flow down the urine because there's so much sugar, so much glucose in the urine, we cannot reabsorb all of it because there is transport maximum. And we have a uniport, that a carrier that moves one type of solute. So if we move only glucose, that's a uniport. It moves one. A symport that can move 2 molecules, like the cingulate. See, the cingulate moves 2, sodium and glucose. That's a symport. And if they are in the same direction, that's a symport co-transport. And then there is the antiport. So one way, so the same carrier throws something out and brings something in. What's an example of that? We just described it. Sodium, potassium, ATPase is the antiport. It's not a symport. It's throwing one outside. Who is the sodium, potassium, ATPase throwing outside? What is sodium, potassium, ATPase throwing outside? Who's the molecule who's getting thrown out, like Black Friday? She's taken from what there is little inside, throw him outside. Who's that? The sodium. And who is it bringing inside? The potassium. And we talked about facilitated diffusion. And if you need ATP, that's called primary. That's the question on the exam. What is facilitated diffusion? Is when you move down concentration gradient and you don't use ATP, but you use another form, kinetic energy. Then there is active transport, where you need a form of energy, and that is known as primary active transport. And in primary active transport, you have ATP, like the sodium, potassium, ATPase. Like here, you see a solute particle enters the channel closes from the top, open from the bottom, throwing it outside. You see this figure? That figure, you have to know it. A solute particle enters the channel of a membrane protein carrier. The solute binds to a receptor on the side and throws it outside. The secondary active transport, you see the cingulate? SGLT. That one is very important because it does not rely on primary active transport, primary active ATP, but it relies on the sodium. And that is created, you see right here. You see, that's your cingulate. See the diagram? Please refer to this diagram of the secondary active transport. The secondary active transport has the green glucose. Do you guys see the green glucose? The green glucose is a lot on the outside. Do you see the orange sodium? So now that upper side is known as the apical side. That's the apical side. You see the square? That's our cell. These are our boundaries. This is the square. Everybody, please look up here. This square, that's our cell. That's our plasma membrane. It has two sides. One side is the apical surface that's got a lot of glucose, and one side is also has a lot of sodium. So it brings them both in. Is that symport or antiport? Symport. They both come in together. When they both come in together, My glucose is gone because I metabolize it. Metabolism of glucose through glycolysis. Then you see there is a lot of orange that piles up. So when there's a lot of orange that piles up, look at what the sodium ATPase does. It uses don't forget that part, it uses ATP in order to throw out sodium from where there is a little bit uphill, not downhill. It throws it against concentration gradient uphill. You see the sodium potassium pump, sodium potassium ATPase is an example of primary active transport. There's a question on the exam. Is the sodium potassium ATPase a primary or secondary? So each pump cycle consumes 1 ATP and exchanges 3 sodium for 2 potassium. So it keeps potassium concentration higher and sodium concentration lower within the cell. It maintains sodium gradient, allowing for secondary active transport, and it regulates the solute concentration and thus the osmosis and the cell volume. It maintains negatively charged resting membrane potential and produces the heat. That's what it does. You have to know this. The sodium potassium ATPase is a form of primary or secondary active transport. Primary. Why primary active? Because you use ATP. If you use ATP, you are primary active transport. And what it uses it, what are the 2 molecules it deals with, the sodium potassium ATPase? Sodium and potassium. Then the sodium potassium ATPase moves sodium from where to where? No, not from out to in. From in to out. Remember, look, we said here, from in, it throws them out. It takes sodium from in to out, where there is a little bit of sodium inside to where there is a lot of sodium outside. Again, it's concentration gradient. How many sodiums are there that are being thrown out? 3. Excellent. The potassium, how many potassium is it being thrown, inside or outside? Is brought in. And how many? 2 or 3? And is it against concentration gradient or with concentration gradient? Is it against concentration gradient or down concentration gradient?
Yeah.
Against it. And the last kind of transport that we're dealing with is not with mediation, is not with a protein. That's vesicle mediated. So in vesicular transport moves large particles and fluid droplets or numerous molecules at once through the membrane. So we have vesicles. These are bubble, like little blebs, little bags. These are bubble-like enclosures of the membrane. And exocytosis brings material into the cell. Endocytosis brings material into the cell. And exocytosis releases material outside the cell. So there are 3 forms of endocytosis. There's phagocytosis, engulfing and destroying large particles, known as cell eating. And there's pseudopods. Pseudopods surround the object and they fuse to form internal phagosome, which merges with the lysosome to form a phagolysosome, within which the object is digested. So the pinocytosis, taking in droplets of the extracellular fluid containing molecules useful in the cell. So the pinocytic vesicles in the cytoplasm, this is known as cell drinking. Pinocytosis is bringing in the substance. Phagocytosis, we talked about the phagosome when we were talking about the pseudopods in our extensions. You see these pseudopods right here. These pseudopods. Yeah, you see the pseudopods? The pseudopods are moving, and that's how the macrophage moves in locomotion. So here, what do we see? We see that the sodium potassium ATPase is an important example of primary active transport. We talked about that. And that is showing you an example of 3 forms of endocytosis, bringing the bubble in, swallowing things up. and that's the phagocytosis. There's also pinocytosis, taking droplets of the extracellular fluid containing molecules using the cell, and pinocytic vesicles. There's a receptor-mediated endocytosis, where particles bind to specific receptors on the plasma membrane, and the pit forms in the membrane. A pit forms in the membrane, and the cytosolic side is covered in clathrin protein and forms clathrin-coated vesicles that is directed to the destination within the cell. Okay, you have to know this figure. This figure is showing you that a phagocytic cell encounters a particle of foreign matter. That's the phagocyte, and that's the purple particle that's dangerous. That could be a microbe, a parasite, bacteria, fungi, and that will be engulfed by the pseudopods. So the pseudopods will go around it, and then the cells surround the particle with its pseudopods, and then the particle is phagocytosed and contained in the phagosome. So the phagosome fuses with the lysosome and becomes a phagolysosome. And the phagolysosome has enzymes from the lysosome that digest the foreign matter. So the phagosome fuses with the plasma membrane. So the indigestible residue is voided by the exocytosis. So receptor-mediated endocytosis. In receptor-mediated endocytosis, the receptor binds. In receptor-mediated endocytosis, the receptor binds, and there's extracellular molecules bind to receptors on the plasma membrane, and receptors cluster together. That's the receptor-mediated endocytosis. So the plasma membrane sinks inward. The plasma membrane sinks inward and forms clathrin-coated pits. Clathrin-coated pits. So the pit separates, forms plasma membrane, forms clathrin-coated pit vesicle containing concentrated molecules from extracellular fluid. Transcytosis is the transport of material across the cell by capturing it on the inside and releasing it on the other side. And exocytosis is when we throw to the outside. So clathrin-coated pit. So we have endocytosis is when, okay, phagocytosis is when we bring in material to the inside of the cell. We are bringing it inward. Endocytosis, bringing inward all of the material. But exocytosis is when we use proteins that fuse with the membrane to throw out all of the content in exocytosis. The cytosol. So now we finish with everything that has to do with the cell membrane. What's in it? How do things enter? How do things exit? Now we're moving to the cytosol. The cytosol is a clear, viscous, watery colloid. What did we say a colloid was when we were differentiating between a solution, a suspension, a colloid, and an emulsion? We said a solution is when you have water dissolving in, when you have little solutes, little tiny solutes like salt, glucose dissolving in water. That's a solution. But when you have big things like red blood cell dissolving in plasma, water, that's not a solution. That's a suspension. When you have proteins dissolving in water, that is known as a colloid. Thank you. And when you have liquid dissolving in another liquid, that is known as an emulsion, like oil vinegar. You know the oil vinegar dressing? Oil vinegar, or maybe fat in breast milk, fat in the breast milk. So the cytosol is a clear, viscous, watery colloid within the cell, and it contains enzymes, other proteins, amino acids, ATP, electrolytes, dissolved gases. So the cytoskeleton is a network of protein filaments. And we talked already about the microfilaments. Where do we find the microfilaments? In the microvilli. Where do we find the microtubules? Microtubules, where do we find them? The cilia, the flagella. So that shows you a cytoskeleton. It has the desmosome that connect them together. It has the secretory vesicles and the microfilaments. Okay, microfilaments, they form a terminal web, and intermediate filaments within the skin cell, and they are made up of protein, keratin. Microtubules, the microtubules are thick and they consist of proteofilaments made up of the protein tubulin. So these are my microtubules. And then we have our organelles. Our organelles are very important to know because some organelles have they all have very important effects. The nucleus, that's usually the largest organelle that is found in the cytoplasm. And most cells have one or few types are anuclear, without a nucleus. What's the best example of a cell that does not have a nucleus? What's the perfect example of a cell that does not have a nucleus? Red blood cell, thank you. Because the red blood cell has no nucleus, red blood cell also has no mitochondria. Because you know, mitochondria use oxidative phosphorylation and aerobic respiration. So it uses oxygen. But who carries red blood cell? Who carries the oxygen? Red blood cell. So imagine you have the red blood cell carrying oxygen and there is mitochondria. The mitochondria will eat up all the oxygen. So that's why the mitochondria, that's why the red blood cell has no mitochondria. The nuclear envelope is a double membrane around the nucleus. It's perforated by nuclear pores. So each formed by a ring of proteins called the nuclear pore complex, and they regulate the molecular traffic through the envelope, and they hold the two membrane layers together. They are supported by a web of protein filaments called the nuclear lamina. The material within the nucleus is called the nucleoplasm. It includes thread-like chromatin, DNA, and proteins, and one or more nuclei where ribosomes are produced. This is the nucleus as seen by the electron microscope. Okay, structure of the nucleus. So these are the ribosomes. We have ribosomes that are embedded. Okay, in the rough endoplasmic reticulum. Why there is a rough endoplasmic reticulum and smooth endoplasmic reticulum? Why? Why is there rough endoplasmic reticulum and smooth endoplasmic reticulum? So why do I have two kinds of endoplasmic reticulum? The rough endoplasmic reticulum is rough because it has a lot of ribosomes embedded on the surface of it, like beads. That's why it looks rough. So there are ribosomes, that right there, you see? That is your rough endoplasmic reticulum with a lot of ribosomes embedded on its surface. But are these the only kinds of ribosomes that I have? There are ribosomes that are free floating in the cytosol, in the cytoplasm. So how many kinds of ribosomes do I have? I have ribosomes on the rough endoplasmic reticulum. They are embedded on my rough endoplasmic reticulum. They are like dots, like beads on the top of the rough endoplasmic reticulum. But are they the only type? Are these ribosomes on the surface of the rough endoplasmic reticulum, are they the only type? No, we have free floating ribosomes in the cytoplasm. What are the two types of ribosomes that we have? One in the it's called free floating in the cytoplasm, and the second one is on the rough endoplasmic reticulum. So what's the difference? Why is that? Why do I have them in two different locations? For no reason. Remember I told you, nothing in life, no reason. There is a reason for everything. So what's the reason why I have ribosomes embedded on the rough endoplasmic reticulum and why I have ribosomes free floating? Okay, remember I told you, whenever you want to know the difference between two things, always begin with what? If you want to know the difference between two things, always begin with the similarities. So whether they are ribosomes on the rough endoplasmic reticulum or ribosomes free floating, in the end, they're going to be making what? So we don't know what do the ribosome make? Proteins. So whether you are a ribosome on the rough endoplasmic reticulum or a ribosome free floating, in the end of the day, you're going to be making proteins. So that's the difference. What kind of protein come out of the rough endoplasmic reticulum associated ribosomes and the free floating ribosome? What's the difference? The difference is if you are a protein who's coming out from the rough endoplasmic reticulum associated ribosomes, you are going to be exported out outside the cell. You're going to be used somewhere else. So the cell make you, but you're not to be used locally. You're going to be used far away. And if you are a protein that will be made by the free floating ribosome, then you are for the own use of the cell. So the cell, remember the cell itself needs protein for its own support in life. You know, the waiter and the chef in the restaurant who make the food for you, they send food to be outside for you, but there's also food inside for them, so they can eat it. Same thing, rough endoplasmic reticulum. You see that endoplasmic reticulum? Any ribosome that is on the surface of the rough endoplasmic reticulum, after she makes her protein, she sends it to the endoplasmic reticulum. The endoplasmic reticulum put a tag on it, known as the signal peptide sequence, like a signal, like a postage stamp. When the rough endoplasmic reticulum put that stamp on the protein, then the cell will know, oh, this is a shipment, this is to be shipped outside, exported outside the cell. But the free floating ribosomes, they are not shipped nowhere, they're just floating inside the cell. We talked about that rough endoplasmic reticulum is filled with ribosomes, and the smooth endoplasmic reticulum has no ribosomes on it. That's why it's so smooth surface. And it's important for detoxifying alcohol and synthesizing lipid and making and storing fats. So that's my endoplasmic reticulum. Rough, that's the rough one, that's the smooth one. Ribosomes, they are the ones responsible for translating. So in the central dogma of biology, we go from DNA into mRNA into proteins. So when we go from mRNA into protein, that's the process known as what? When we go from DNA into mRNA, that is known as transcription. But when we go from mRNA into protein, that's known as translation. The Golgi complex is like pita bread, on top of each other. But it's very important. It's a system of membranous cisterns that synthesize carbohydrates and modifies the newly synthesized proteins. It is a secretory vesicle. You see, it looks like pita breads or stuck up on the top of each other. And then there's the lysosomes. They are very important because inside them they have granules. They have metalloproteinases, degradative enzymes, acid that burns. So when the lysosome, that's why when we have an inflammation, is because the lysosome leak out to the nearby tissue, destroying and damaging, causing redness, heat, pain, swelling. These are the lysosomes that are supposed to be used to destroy the invading the infectious microbe. But sometimes they can destroy the cell itself. So the lysosome is where you have your knives, is where you have the stuff that cuts and destroys. So you see in autophagy, that is the digestion of the cell's surplus organelles. Peroxisomes. Peroxisomes from the word peroxide. We love peroxide. Peroxide is so good. So what they do is sometimes we have free radicals. I talked to you about people who drink the alcohol. The alcohol is going to be metabolized in the body to acetaldehyde with free radicals causing oxidative stress, damaging the liver, alcoholic liver disease. That problem now, when there is moderate amount of stress, what happens is the peroxisome is going to try to take that reactive oxidative species and mute it and give us something that is useful, known as hydrogen peroxide. So the function is to use molecular oxygen to oxidize our organic molecules, and the reaction produces for us hydrogen peroxide. There's another enzyme known as catalase. It breaks down this hydrogen peroxide into water. So catalase, we take down the step even further. So there's an enzyme known as SOD. SOD known as super. We talked about that last week. Superoxide dismutase. And superoxide dismutase converts for us reactive oxygen into, converts for us superoxide into regular oxide, oxygen with hydrogen peroxide. Then hydrogen peroxide catalase will come in and convert hydrogen peroxide into water plus oxygen that we can breathe. So we are trying to mute things down. Proteasomes. Proteasome is like a shredder, but it's a shredder of the proteins. So when there is a misfolded protein, you know, we talked earlier about proteins folding on top of each other. How do proteins fold up on top of each other to go from primary structure to secondary structure to tertiary structure to quaternary structure? How do they do that? How do the proteins fold up from primary structure to secondary structure to tertiary structure to quaternary structure? What is the folding?
An hour ago?
Mm-hmm. Mm-hmm. So who causes the folding? The hydrogen bonding, right? The hydrogen bonding is what causes them to fold up on the top of each other. Sometimes we have misfolded proteins, improperly folded proteins, misfolded, they're not folded properly, misfolded proteins, improperly folded proteins. So these properly folded proteins, if we don't get rid of them, what's going to happen? They're going to pile up on our brain, giving us what? Mad cow disease. So mad cow disease is when you have improperly folded protein, and people who smoke coke, you know cocaine? Who do the cocaine, they have a lot of misfolded proteins in the brain, but they have mad cow disease. Now, the proteasome, usually, if you are a normal person, you don't do those kind of substance abuse, drug dependence. What happens to you now is that if you have some misfolded proteins, the shredder of the proteins is known as proteasome. So the proteasome will any misfolded protein that is damaged, improperly folded, it will have a purple tag. The tag is called ubiquitin tag. Ubiquitin tag. And every tag that has a ubiquitin, the proteasome will go shred it, swallow it, destroy it, cut it up to pieces. That's the proteasome. You see, that's the proteasome. That's the purple tag. That's the targeted, misfolded, bad protein. The tag will bind to it, and then the proteasome will shred it and cut it to small pieces that cannot cause mad cow disease. The mitochondria, we know from high school that it is the powerhouse of the cell. It provides energy for the cell, but it is also responsible. I believe there's a question on the exam that asks you what is responsible for the cell death. We talked about that in the mitochondria, you have the programmed cell death. If a cell has many improper proteins that are defective or damaged, or maybe there's an infection or severe oxidative stress. The cell has been damaged beyond salvage. The cell needs to be removed so another cell can be on the top, on its place on top of it. What happens is the cell needs to die, but it's called programmed cell death. So it dies by disposing of itself, getting rid of itself, properly disposing of itself, and getting out from that medium through a programmed way. Who does that programmed way of killing the cell? Known as apoptosis. 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 Programmed cell apoptosis. That is known as programmed cell death. Who does that? Apoptosis. Programmed cell death. The mitochondria. Programmed cell death. Programmed cell death. That's the mitochondria. The centrioles are important. They are a short cylindrical assembly of microtubules, and two centrioles lie perpendicular to each other within the centrosome and from the basal bodies of the cilia and the flagella. Then there's inclusions. Inclusions are like bags. These are bags that store the products of foreign matter. We began talking about the structure of the cell, and now we have accumulations of, you can have, there could be full of fat, they could be full of cholesterol, they could be full of proteins. They are inclusions, so they are inside and they are closed inside. That's why they call them inclusions. They could also, they don't only be inclusion of good things, they could be inclusion of negative or bad things. They could be inclusions of accumulated cell products for foreign bodies like the virus, intercellular bacteria, and they are never enclosed in a unit membrane. They are not essential for survival. Okay, does anybody have a question? Anybody has a question? Or are we done? 3.